Hey, ungerminated peas. We know you're starting to grow up and your body's starting to change and develop. It happened to your grandparents, it happened to your parents, and it's even starting to happen to some of your friends. Now it's time for you to make some glucose of your own. Let me enlighten you on your next stage of life. Without further ado, here's Photosynthesis 101. Let's start with a broad overview. Photosynthesis is the process where plants convert light energy into chemical energy. Here are some key terms that will help you thoroughly understand both the light reactions and dark reactions processes. Autotrophs, that's you. You are organisms that use light as a source of energy to produce your own food, making you photoautotrophs. Chloroplasts, this is where photosynthesis occurs. They are organelles in your cells that are located mostly in the mesophyll of the interior leaf and contain the thylakoids and stroma, which are key locations in photosynthesis. Thylakoids. These are interconnected membranous sacs that separate the stroma from the thylakoid space and contain chlorophyll. The light reactions take place here. Stroma. This is the dense fluid that fills the chloroplast's inner membrane. It is also the location hosting dark reactions, aka the Calvin cycle. Try to remember the definitions of these words. As your teachers, we will be referring to them throughout the class. Keep them in mind. Here is a visual of this broad overview. As you can see, the light reactions take place in the thylakoid membranes. Overall, they produce ATP and NADPH, which is the chemical energy that is sent to the Calvin cycle. The light reactions also release oxygen gas as a waste product. These players are crucial in the Calvin cycle's production of sugar. Now let's take a closer look at the light reactions, starting with photosystem 2. Step 1. Photosystem 2. Photosystem 2 is found in the thylakoid membrane. It contains pigment molecules that absorb photons of light. As light enters the chloroplast, it is transferred among the pigment molecules to excited chlorophyll molecules called the P680 that transfer the electron to a primary acceptor. As this is happening, water is split into H plus and oxygen, and the electrons from this process go into the P680 chlorophyll while it is transferring electrons to the acceptor. Step 2. The electron chain. Each photoexcited electron passes from photosystem 2 to photosystem 1 via an electron transport chain. This is also located in the thylakoid membrane and is composed of several proteins. The exergonic fall of electrons provides energy for ATP synthesis via the mechanism of chemiosmosis, which we'll be talking about next. Step 3. Chemiosmosis. Chemiosmosis drives ATP synthesis using the potential energy of a concentration gradient across the membrane. But how is this concentration gradient generated? As photoexcited electrons are passed down the electron transport chain connecting the two photosystems, hydrogen ions are pumped across the membrane from the stroma into the thylakoid space, generating a concentration gradient as shown. The movement of hydrogen ions down their concentration gradient allow ATP synthase to produce ATP. Step 4. Photosystem 1. In photosystem 1, the light energy excites an electron of chlorophyll P700 in the reaction center. The primary electron acceptor captures the excited electron, and an electron from the bottom of the electron transport chain replaces the lost electron to P700. The excited electron of photosystem 1 is passed through a short electron transport chain to NADP+ reductase, an enzyme that reduces it to NADPH. Now that we've gone over the light reactions, we can move on to the dark reactions. The products of the light reactions, NADPH and ATP, will be used up in the Calvin cycle. Welcome to the Calvin cycle, aka the dark reaction. Now we're going to take the ATP and NADPH that we generated in the light reactions, along with some carbon dioxide from the surrounding air, and use them to achieve our ultimate goal, to make G3P, aka glyceraldehyde 3-phosphate. G3P is important because plant cells need G3P to make glucose and other organic molecules. The Calvin cycle occurs in the stroma of the plant cell's chloroplast. The dark reaction can be broken up into three main parts: carbon fixation, reduction, and regeneration of RUBP. This is the bigger picture. As you can see, one step leads to the next, like a cycle. Now let's take a closer look at what's actually happening in the process. Step 1, carbon fixation. In the first step, we've got the enzyme rubisco, carbon dioxide, and RUBP, the 5-carbon sugar. Rubisco helps carbon dioxide, the carbon specifically, and RUBP combine. They create a very unstable product. The product then stabilizes by splitting into 2 molecules of the 3-carbon organic acid known as 3-PGA. Step 1 is complete. Step 2, reduction. In the second step, each 3-PGA receives a phosphate group from the ATP and forms 1,3-bisphosphoglycerate. NADPH then enters the scene and reduces 1,3-biphosphoglycerate to 6-G3P. There is only a net output of 1 3-G3P because the other 5-G3P continue on throughout the cycle. 3. Regeneration of RUBP. In this phase, 3 more ATP are used to transform the 5 remaining 3G3P into 3 molecules of RUBP. Yay! We have RUBP again. RUBP is ready to receive more carbon dioxide to restart the cycle. Here's a basic overview. The light reactions uses water to make ATP and NADPH, which the Calvin cycle uses to make G3P. The Calvin cycle then recycles ADP and NADP+ back to the light reactions so it can continue the cycle.
Thank you for taking Photosynthesis 101. We hope we prepared you for the next stages in your life.
You are all such bright students.
See you next time!